This is the first lesson of Chapter 7 on cellular respiration and fermentation. In this lesson, we're going to look at an overview of the process of aerobic cellular respiration, something that we'll get into greater detail in the subsequent lessons of this chapter. So let's start off with an understanding of what is cellular respiration and why is it important to a living organism. One of the requirements for life is that organisms need to be able to obtain energy from outside sources. Some organisms, like the marmot shown here, have to consume food, and from that food, they're able to harness energy that's stored within that food in the form of the sugars. Other organisms, like the plant itself that the marmot is eating, gets its original source of energy from the sun, which then converts it into a form of food for the plant to be able to use. But regardless of whether the animal, like the marmot, needs to consume food or the organism can make its own food, they still need to be able to get that energy from outside of their body. Once that energy is in their body, it's in the form of potential energy. And so now we're going to look at how that potential energy in the form of the food can be then used by the cells to do work. Recall from chapter 6, we learned about some introductory metabolism concepts, including catabolic and anabolic pathways. In this scenario, we're going to be looking specifically at the catabolic pathways or destructive pathways that take large, more complex molecules, break those down into smaller, simpler molecules, and through that process, yield energy through the oxidation of those organic fuels or organic molecules like glucose, for example. But glucose is not the only food that can be used as a source of energy for cellular respiration. So even though we're going to be looking at glucose as our kind of representative food molecule, be familiar with the fact that the other organic molecules, including carbohydrates, other carbohydrates, not just glucose, proteins, and fats, can also contain energy in the form of potential energy as food to be used in cellular respiration. So as I mentioned in the beginning of this lesson, we're going to be doing an overview of the whole process right now of aerobic cellular respiration. And we will get into the greater depths of this with future lessons. So for now, let's start with the overall reaction. The balanced chemical equation for cellular respiration is shown here. We have glucose, C6H12O6, oxygen as reactants, and the products are carbon dioxide, water, and energy in the form of ATP and heat. The whole purpose of cellular respiration is to get that energy specifically in the form of ATP. It starts with the process of glycolysis. Glycolysis is the breakdown of the glucose into two molecules of pyruvate. Next is pyruvate oxidation. Then the citric acid cycle, which is where the breakdown of glucose is completed. And finally, oxidative phosphorylation, which is where the big payout of ATP synthesis occurs. We have also a visual roadmap of cellular respiration, and this is one of the concepts that really lends itself nicely to visual representations, models. So you'll find that you have a copy of this in your course packet. It might be that you need to fill in some missing terms. And so I would highly encourage you to do that while you're following along with the lesson. And also use this as a tool to test yourself. See if you can draw out your own diagrams. Flashcards are good for certain concepts, but you have to try other tools to try to understand and remember these other more complex processes like cellular respiration and also photosynthesis coming up next chapter. So the same 4 stages are shown here, but now you can see like a roadmap and spatial locations of where this is happening. So let's identify where we are in the organism that this is occurring. Okay, so imagine all of this outer peachy colored is representing the inside of a cell of a eukaryote. So that would be the cytoplasm. Everything basically aside from this structure here, which is the mitochondria. We'll get into that later. But for now, we're looking at the fact that glycolysis occurs in the cytoplasm. And it begins with the breaking of 1 molecule of glucose into 2 molecules of pyruvate. Through glycolysis, there is going to be a little bit of ATP formed through a process called substrate level phosphorylation. Maybe you remember learning a bit about substrates in our last chapter with enzymes. So this is referring to the fact that there is an enzyme that's responsible for adding a phosphate group from a substrate, we're just keeping it generic right now, to ADP to ultimately produce ATP and a byproduct. So this is a mechanism through which ATP is made in glycolysis. We're going to see another type of way that ATP is made in a later stage of cellular respiration. Okay, but the point really here is, well, twofold, I guess. It depends on the organism. If this is a eukaryotic organism, then we're going to continue following the pyruvate into the mitochondria. It is good that some ATP is made here, so that's another beneficial outcome of glycolysis, but it's really not like a big payout of ATP. It's actually only 2 ATP per 1 glucose, which you'll find out in the end of this that that's really not that much. So just keep that in mind that there's ATP coming from glycolysis, but let's continue following the flow of the food, if you will, which is now pyruvate, into the mitochondria. So that's what this structure is here, and here's on the right more of a detailed representation of the mitochondria. It has a double membrane, so it's a membrane-bound organelle that has 2 membranes, an outer and an inner membrane. And notice that the inner membrane has these like enfoldings, or cristae is what those are called. And within the mitochondria inside, like the innermost place, is the matrix. That's kind of like cytoplasm. It's all the fluid and everything else inside of the innermost part of the mitochondria. Now, what's most relevant about the mitochondria and cellular respiration is that ATP is synthesized on those cristae, on those inner membranes. Remember that form and function go hand in hand in biology. So the more enfoldings, the more potential it has to make ATP. And we'll see that later. So right now, we're seeing how the pyruvate is being shuttled into the mitochondria, and through this process of pyruvate oxidation, electrons are being stripped from the pyruvate through this oxidation process and becoming ultimately a compound called acetyl-coenzyme A, or acetyl-CoA for short. Now that acetyl-CoA is what feeds the next step of aerobic cellular respiration, the citric acid cycle. Now this is occurring in the matrix of the mitochondria, and this is where the complete breakdown of glucose takes place. So we started with glucose, and then that got oxidized to pyruvate, which got oxidized to acetyl-CoA, and now that carbon compound that was once glucose is completely stripped of its electrons, and there will really be no more food after the citric acid cycle. Similar to glycolysis, some ATP is also formed in the citric acid cycle by substrate level phosphorylation. So like before, an enzyme transfers a phosphate group from an inorganic from an organic substrate to ADP, forming ATP. So there's a little bit of ATP, but again, it's not the big payout that we're going to see in the final step. We will study the citric acid cycle in much greater detail, but I do want to just give you a little preview of that. This is a condensed version of the citric acid cycle. You will need to know like what comes into the cycle and what comes out of the cycle, the major compounds. So those are highlighted here. For every one acetyl coenzyme A that feeds the citric acid cycle, 2 carbon dioxide molecules are given off. Notice that those are the same 2 gray balls that came in. So all the carbons that feed the citric acid cycle are going to be ultimately broken down and released as CO2. So you might be wondering, well, what's the point of the citric acid cycle? Is there going to be any energy yield from this? The answer is yes. The energy is going to be contained within these compounds, NADH, FADH2, which carry electrons ultimately to the last stage, oxidative phosphorylation, and then also ATP here, the 2 ATP that we talked about already. Okay, but we're now going to look at what the other form of those energy molecules are, the NADH and the FADH2. These electron carriers are going to shuttle electrons taken from these earlier stages to the final stage of aerobic cellular respiration, which takes place on the inner membrane of the mitochondria. And here there's going to be a coupling between these 2 processes called electron transport and chemiosmosis. Now we will have to dive really deep into that in our future lessons. For now, I just want you to know like where we are in the cell, where these processes happen, and what's really the point and the big outcome. And so for oxidative phosphorylation, we're going to get a huge payout here of ATP, about 26 or 28 ATP, compared to the 2 from glycolysis and the 2 from the citric acid cycle. That's a lot of ATP. So in total, for one glucose molecule that feeds the whole process here, there could be up to 32 ATP made. Each of those molecules of ATP has 7.3 kilocalorie per mole of free energy that would be energy available to do work. So if you multiply 7.3 times 32, we're not going to have to do that, but just you could think about how much free energy that is for every one glucose molecule. Okay, so again, this is meant to be an overview just to kind of introduce you to some of the major steps and major reactants and products and what the whole point of aerobic cellular respiration is. So in our future lessons, get ready to get into more detail, but definitely be sure that you have a basic understanding from this introductory lesson.